students. Today we are going to learn the second part of the lesson number 4, Principles of Inheritance and Variation. In the first half, we have learned about the important concepts like multiple alleles, and that multiple alleles ABO blood type we have learned, and we have learned about the RH factor. In that we have learned the Fisher and Race hypothesis, Wiener's hypothesis, incompatibility of the RH factor, that is erythroblastosis fetalis, and we have also learned about the sex determination in the last class. Today we are going to learn other important concepts like sex-linked inheritance. What is meant by sex-linked inheritance? What is inheritance? Inheritance means we get the characteristic features from our parents. Both the male and the female parent pass on the characteristic features through their genes. And this inheritance of the gene or the trait that is determined by the gene located on one of the sex chromosome. If that particular gene is located on one of the sex chromosome, then it is called as sex-linked inheritance. You know what are sex chromosomes? So every human being have got 23 pairs of chromosome. Out of this 23, first 22 pairs are called autosomes, and the last pair is called as sex chromosome. We call it as XX in female and XY in male. So the genes are present on the differential region of the X and Y chromosomes are called sex-linked genes. So what do you mean by differential region? This differential region will not be present in the other chromosome. There is no corresponding gene in the other chromosome. They are specially present only on the X and Y chromosome. So these genes present on the differential region of the X and Y chromosomes are called as sex-linked genes, and the inheritance of these sex-linked genes is called as sex-linked inheritance. The genes present in the differential region of X chromosome are called X-linked genes, and the X-linked genes have no corresponding alleles in the Y chromosome, because in male only one X is present and one Y will be present, and so the X-linked genes have no corresponding alleles in the Y chromosome. The gene present in the differential region of the Y chromosome are called as Y-linked genes, and these genes are otherwise called as Hollandric genes. I repeat, the genes that are present in the differential region of the Y chromosome are called as Y-linked or Hollandric genes. The Y-linked genes have no corresponding allele in the X chromosome. So both are different. They will not be having any corresponding genes. X will not have corresponding gene in the Y, and Y will not have any corresponding genes alleles in the X chromosome. So the Y-linked genes inherit along with Y chromosome only. So only the males will be getting this genes, and so they are phenotypically expressed only in the male sex, where the Y chromosome is passed on. The sex-linked inherited traits are more common in males than in females. Why? Because males are only becoming hemizygous. So hemizygous means they will not be having the corresponding allele in the X chromosome. And so most of the sex-linked traits will be common in male only, because males are hemizygous, and therefore they express the trait when they inherit one mutant allele. The X-linked and the Y-linked genes in the differential region, that means non-homologous region, do not undergo pairing or crossing over during meiosis. So the genes which are present in the differential region of the X as well as in the Y region will not get mixed or exchanged during the crossing over during meiosis. So the inheritance of the X and the Y-linked genes is called as X-linked inheritance. Now let us learn what is homozygous, heterozygous and hemizygous. What do you mean by homozygous? A cell is said to be homozygous for a particular gene. For example, if we take this B, capital B gene, particular gene, when identical alleles of the gene is present on both the homologous chromosome. This is the homologous chromosome. So B, here also B present, capital B. Same way, here another homologous chromosome, small b is present. So a cell is said to be homozygous for a particular gene when identical alleles of the genes are present on both the homologous chromosome. So the cell or the organism is said to be homozygote. Whereas in heterozygous condition, capital B and small b in the another homologous chromosome. So a diploid organism is heterozygous at a gene locus when its cell contain two different alleles, one wild type and one mutant type. The small b is called as mutant type. So mutant allele is present. The cell or the organism is said to be called as heterozygote. So same way here also you can see. A special condition which is called as hemizygous is present in male, where the male has an abnormal allele for the gene located on the X chromosome and there is no other copy of that particular gene is present in the Y chromosome. So he is neither homozygous or heterozygous, he is referred as hemizygous. It is present only in one of the chromosome and no copy of it is either the mutant or the wild type is not present in the next homologous chromosome. Then this condition is said to be called as hemizygous. Now let us learn the inheritance of X-linked genes. Red-green color blindness, which is otherwise called as daltonism. Daltonism, the person will not be able to distinguish between red and green. And that condition is called as daltonism or red-green color blindness. The next condition is called hemophilia and Duchenne muscular dystrophy are the examples of X-linked inheritance in humans. So three are listed in your book. Red-green color blindness or daltonism, hemophilia and Duchenne muscular dystrophy or X-linked inheritance. First let us learn hemophilia. What is hemophilia? It is commonly known as bleeder's disease. This is very common in men than in women. And this hereditary disease was first reported by John Cato in 1803 and it is caused by a recessive X-linked gene. When a person tends to carry a recessive gene for this hemophilia, will lack normal clotting substance which is known as thromboplastin. So thromboplastin is essential for clotting of the blood. Hence, even in minor injuries, they will bleed continuously and which may also lead to death. Females become the carrier of the disease and they will be able to transmit the disease to 50% of their sons, even if the male parent is normal. And so hemophilia follows a crisscross pattern of inheritance. What do you mean by crisscross pattern? So it is transmitted from the mother to the son and from son to the granddaughter. So it is passed on from female to male, male to female and from female, like that it is going on. So it is called as crisscross pattern of inheritance. Next we have the color blindness. So in human beings, a dominant X-linked gene is necessary for the formation of the color sensitive cells called cones. So we know the retina will have two types of cells which is called rods and cones will determine the colors. And the cones are formed because of the genes which is present in the X chromosome, X-linked gene. The recessive form of this particular gene is incapable of producing the color sensitive cone cells. If the color sensitive cone cells are not produced, then the person becomes color blinded. Homozygous recessive female, so which is determined, which is expressed as XCXC. So the small c is for the recessive gene of the color blind. So the homozygous condition, females XCXC and the hemizygous recessive males are unable to distinguish the red and green color. So they both will not be able to see will not be able to distinguish red and green color. So the inheritance of the color blindness can be studied in two types of marriages. Let us see one by one. So a marriage between color blind man. So you can see XCY. And a normal visioned woman, so you can see XX. X plus means there is no colorblind gene allele in the women, normal women. Will produce normal visioned male and female individuals in the F1 generation. So F1 generation, first generation, both the male and the female, the son and the daughter will be normal. But the females will be carriers and they will pass on these genes to the next generation. So here can we see the gametes produced by the colorblind man will be XC and Y. Here normal XX gametes. So when this is fertilized, when one egg is fertilized by this colorblind gene, then the individual produced will be XCX plus, who will be a daughter, and here also XCX plus, who will be a daughter, and both will become the carriers of the C colorblind gene. Here the sons will be normal because here in Y the colorblind man is only contributing the Y gamete and which will not have the allele for the color blindness. So the sons who are produced will be normal. So the marriage between the F1 normal, so F1 generation normal visioned women with normal visioned male. So F1 generation women, carrier daughter we have seen, isn't that? That carrier daughter, if she is marrying a normal man, then the gametes are XC and X plus, and here X plus and Y, normal gametes. When this particular egg is fertilized by the X chromosome, then the individual produced will be a carrier daughter. Then when this egg is fertilized by this Y gamete, then the person who is produced will be a color blind son, because even in the hemizygous condition the males will be color blind whereas the females will be carrier. So colorblind trait inherited from the male parent to his grandson through the carrier daughter. So this is an example of crisscross pattern, from father to daughter carrier and from daughter to the grandson. So this pattern is called as crisscross inheritance, pattern of inheritance in hemophilia it is seen. Now the next type of inheritance, that is the marriage between the normal visioned man and color blind woman. We all well know that in the hemizygous condition itself the man will become color blind, whereas only in the homozygous condition the woman becomes the color blind. In hemizygous condition women becomes carrier. Now if the color blind women means both the genes, both the alleles must be a recessive allele for color blind. So XC when she marries a normal visioned man XY, the all F1 sons will be color blind but the daughters will be normal because they all become carrier in the hemizygous condition. So let us see the pattern of inheritance here. So XC color blind women and a normal man, they produce the gametes XC and X and Y. So when this X are fertilized by this X and Y, the female's daughter will become a carrier, but they are normal daughters, but they will become the carrier of the gene, whereas sons become color blind because both the X produced are having the color blind allele, and so the sons will be in hemizygous condition itself color blind. So in F1 generation it produces normal daughter but carriers and color blind sons. Whereas in the F2 generation, when this normal daughter, carrier daughter, marrying a person who is color blind, then the gametes produced will be XC and X plus here and XCY gametes. So in the pattern of inheritance, when this egg is fertilized by this sperm, XC sperm, then the individual daughter would be a color blind daughter and one daughter would be carrier daughter. So same way one colorblind son as well as normal son are also produced in the next type of inheritance. Now let us see the inheritance of the Y-linked genes. So genes in the non-homologous region of the Y chromosomes are inherited directly from male to male because Y chromosome is passed on from male to male only. So in human beings the Y-linked or the Hollandric genes for hypertrichosis. Hypertrichosis means excessive development of the hair on the pinna of the ear. So you can see in the picture. This is transmitted directly from the father to the son because male only inherit the Y chromosome. And so females inherit only X chromosome, so they will not be getting this particular gene from the father, and they are not affected also. So what is meant by karyotyping? So karyotyping is a technique through which a complete set of chromosome is separated from a cell. So the whole set of 23 set of chromosomes we say for human beings. So complete set of chromosomes are separated from a cell, and these chromosomes are arranged in pairs and it is studied. And this is what is called as karyotyping. What do you mean by ideogram? An ideogram refers to the diagrammatic representation of these chromosomes. It is not the actual picture, but it is the diagrammatic representation of the chromosomes. Now preparation of the karyotype. To learn the chromosomes, karyotype is very essential, and it was first done by Geo and Levin in 1916. They described the simple method of culturing the lymphocyte. So lymphocytes are the WBC from the human blood, and during the mitosis, the mitosis actually is induced in these cells, and it will be followed by the addition of colchicine, a substance which will arrest the cell division of the cell in the metaphase stage, and this metaphase stage is suitable for separating all the chromosomes, and on a suitable spread of metaphase chromosome is photographed. So the individual chromosomes are cut from the photograph and they are arranged in orderly fashion in homologous pairs and studied. What is meant by karyotype? Photographic arrangement of the homologous pairs of chromosome is called as karyotype. This you can see in this picture. So chromosome banding permits the structural definition as well as the differentiation of the chromosome. Everything can be studied, observed and studied from this karyotyping. Now applications of karyotyping. It helps to find the gender, whether it is male or female. You can see here X and Y chromosome, we will be able to understand the cell is taken from the male or the female. So it helps to understand the gender identification. It is used to detect the chromosomal aberration. Aberration means malformation like deletion, duplication, translocation, non-disjunction. So anywhere it has cut and joined somewhere else like that. So all the aberrations can be studied using this karyotyping. It also helps to identify the abnormalities of the chromosomes like aneuploidy, excess number of chromosomes or the less number of chromosomes. All these can be studied, and it is also used in predicting the evolutionary relationship between the species. Genetic diseases in human beings can be detected by this technique. So genetic diseases in human beings can be detected by studying the karyotyping. Human karyotype. Depending on the position of the centromere, this red spot is called the centromere, the relative length of the two arms. So these are the arms of the chromosome. So the chromosomes can be classified into three types like metacentric, submetacentric, acrocentric. So and also telocentric. When the centromere is in one top corner, we call it as telocentric end. So when it is present in the middle, we call it as metacentric. Less than half, we call it as submetacentric, and acrocentric when it is near to the head and tail region like that. So it is called as acrocentric. The photograph of the chromosomes are arranged in order of the descending length in the group from A to G. Next we have pedigree analysis. Pedigree means family tree. Pedigree or family tree is drawn with standard genetic symbols showing the inheritance pathway for the specific phenotypic character. So here you can see the symbols and the explanation. So square will determine male, circle for female, and this is mating or marriage, and they produce the daughter and the son, one boy and one girl in order, and this is the twins produced by dizygotic, and here monozygotic with the same zygote, two daughters or sons are produced, twins are produced. Then here sex unspecified, and here the number of children and the sex. The square determines the male and the circle determines the female. Here you can see the affected individuals. If you are studying about any disease, if this is fully shaded, then both the individuals are affected. If it is partly shaded, then they will be heterozygotes, autosomal recessive. So they will be carrier and they will be passing on like that. Next we shall learn about the genetic disorders. What is a genetic disorder? A genetic disorder is a disease or a syndrome that is caused by an abnormality in an individual's DNA. So abnormalities can range from a small mutation in a single gene to the addition or subtraction of the entire chromosome or even a set of chromosomes. So it can range from single gene to a set of chromosomes. So genetic disorders are of two types, namely Mendelian disorder and chromosomal disorder. The first one is Mendelian disorder. So alteration or mutation in a single gene causes the Mendelian disorder, and it can be transmitted from the parents to the offsprings in the same line, and so it is called as Mendelian pattern of inheritance. And some of the examples of Mendelian disorders are Thalassemia, Phenylketonuria, Albinism, Huntington's chorea, sickle cell anemia, etc. And these disorders may be dominant or recessive, or it can be present in the autosomal chromosome or sex-linked chromosome. So Mendelian disorder, here you can see the diagrammatic representation. Affected father, normal mother. So they have the DN, the chromosome DN, and here NN, normal. So D for affected. So they may produce affected female, normal male, affected male, and normal female. So a genetic disease showing Mendelian pattern of inheritance caused by a single mutation in the structure of DNA is known as Mendelian disorder. So definition for Mendelian disorder: a genetic disease showing a Mendelian pattern of inheritance and caused by a single mutation in the structure of DNA. This is called as Mendelian disorder, and such disease trait can be dominant or recessive also. Thalassemia. So thalassemia is an autosomal recessive disorder. Autosomal means other chromosomes other than the sex chromosome, and it is caused by a recessive gene. It is caused by the gene mutation resulting in the excessive destruction of RBCs due to the formation of abnormal hemoglobin molecules. So destruction of RBC takes place because of the abnormal hemoglobin molecules. Normally hemoglobin is composed of 4 polypeptide chains, 2 alpha chain and 2 beta globin chains. Thalassemia patients have defects in either the alpha or the beta globin chains, causing the production of abnormal hemoglobin molecule, resulting in anemia. Thalassemia can be classified into alpha and beta based on which chain the hemoglobin molecule is affected. So alpha thalassemia, it is controlled by two closely linked genes like HbA1 and HbA2 on the chromosome number 16. The mutation or deletion of one or more of the 4 alpha gene alleles causes alpha thalassemia. Beta thalassemia, the production of beta globin chain affected. It is controlled by a single gene HBB on the chromosome number 11, and it is the most common type of thalassemia, and it is also known as Cooley's anemia. In this disorder, the alpha chain production is increased and damages the membrane of the RBC. The next disease is called phenylketonuria, and phenylketonuria is an inborn error of phenylalanine metabolism, and this is caused due to the presence of a pair of autosomal recessive allele. The recessive allele name is called PA gene. This is present in the chromosome number 12, and this gene is responsible for the synthesis of the hepatic enzyme called phenylalanine hydroxylase. If this enzyme is not produced, because this enzyme is essential for the conversion of phenylalanine to tyrosine. If the individual does not produce this enzyme, the phenylalanine will get accumulated in the cell and it gets converted into phenylpyruvic acid and other derivatives. It is characterized by severe mental retardation outside and light pigmentation of the skin and the hair. And also they will excrete phenylpyruvic acid in their urine. So phenylalanine is converted into tyrosine only in the presence of phenylalanine hydroxylase. When this enzyme is not produced, it is converted into phenylpyruvic acid and other derivatives, causing the disease called phenylketonuria. Next we have albinism. Albinism is also an inborn error of metabolism caused due to an autosomal recessive gene. So you can see the picture of a person who is having albinism. So melanin pigment is responsible for the skin color. So absence of melanin causes a condition called albinism. A person with the recessive allele will lack the tyrosinase enzyme system, so which is required for conversion of dihydroxyphenylalanine, in short we call DOPA, into melanin. So tyrosinase helps to convert dihydroxyphenylalanine 3,4-dihydroxyphenylalanine DOPA into melanin. Melanin is the pigment present inside the melanocytes. In an albino, the melanocytes are present in the normal number in their skin and hair, iris, etc. but lack this pigment. The pigment will not be present. The next disease is called Huntington's chorea. So this is also an inherited disease by an autosomal dominant lethal gene in man. It is characterized by the involuntary jerking of the body and progressive degeneration of the nervous system, which is accompanied by the mental and physical deterioration. The patient normally will die off between the age of 35 to 40. Chromosomal abnormalities. So every human beings in diploid cell will have 46 chromosomes, that is 23 pairs. The chromosomal disorders are caused by the error in the number as well as the structure of the chromosomes. Chromosome abnormalities are usually occurring when there is an error in the cell division. So failure of the chromatids to segregate during the cell division resulting in the gain or loss of one or more chromosomes is called as aneuploidy. So definition for aneuploidy, very important. Failure of the chromatid to segregate during the cell division resulting in the gain or loss of one or more chromosomes is called as aneuploidy. And this is caused by the non-disjunction of the chromosome. Chromosomes will not separate properly. So group of signs and symptoms that occur together that will characterize a particular abnormality is called as syndrome. So in human, Down syndrome, Turner syndrome, Klinefelter syndrome, Patter's syndrome are some of the examples of the chromosomal disorder. So here non-disjunction, meiosis 1, meiosis 2, when the homologous chromosomes fail to separate from each other causes non-disjunction. Down syndrome or trisomy 21. Trisomy 21, that is 21st chromosome will have 3 chromosomes. Instead of a pair of chromosome, one chromosome will be extra. So trisomic condition of chromosome 21 result in Down syndrome. It is characterized by severe mental retardation, defective development of the central nervous system, increased separation between the eyes. You can see the space between the eyes, flattened nose, ears are malformed, mouth is constantly open and tongue will protrude. See here, the 21st pair will have the 3 chromosomes which is in this Down syndrome. The next one is Patter's syndromes or trisomy 13. So 13th chromosome will have 3 chromosomes which is called as trisomy condition of chromosome 13, Patter's syndrome. So meiotic non-disjunction is thought to be the cause for this chromosomal abnormalities. So it is characterized by the multiple and severe body malformation as well as profound mental defect or deficiency. So small head with small eyes, cleft palate, malformation of the brain and internal organs are some of the symptoms of this syndrome. Allosomal abnormalities. So meiotic and mitotic non-disjunction of sex chromosome causes allosomal abnormalities. So several sex chromosomal abnormalities have been detected, example of which is Klinefelter syndromes and Turner syndrome. The first allosomal abnormality in human beings is Klinefelter syndrome, that is XXY males. In this genetic disorder, the individual will have one extra X chromosome resulting in the choriotype of 47 chromosomes. The person with this syndrome will have 47 chromosomes, that is 44 autosomes and XXY allosomes. These individuals are usually sterile males, tall, obese with long limbs, high-pitched voice, underdeveloped genitalia and have feeble breast development. Here you can see the XXY sex chromosomes, Klinefelter syndrome. Next syndrome is called Turner syndrome, that is X0 females. In this genetic disorder is due to the loss of one X chromosome in the female, and so the resulting choriotype will have only 45 chromosome, one less. The person with this syndrome will have 45 chromosomes, that is 44 autosomes and 1 X chromosome, and they become sterile females. The characteristic features of low stature, webbed neck, underdeveloped breast, rudimentary gonads, lack of menstrual cycle during the puberty are the main symptoms of this syndrome. and here you can see loss of one X chromosome leads to the Turner syndrome. Next we have the concepts of eugenics. Eugenics is the term from Greek. Eu means true or good, well. So well born. It is a set of beliefs and practices that aims at improving the genetic quality of the human population, which is called eugenics. What is eugenics? So eugenics deals with the improvement of the human race by genetic engineering. So by changing the genes, the appearance or the phenotype of the individual also can improve. So eugenics, which literally means good appearance and the normal appearance, and it is the science making phenotype improvement to the human after birth, generally to the affect a problematic genetic condition. What is meant by euthenics? Euthenics is the study of improvement of human functioning and well-being by the improvement of the living condition or environment, affecting the improvement through altering the external factors such as education and the controllable environment, including the prevention and removal of the contagious diseases and parasites, environmentalism, education regarding employment, home economics, sanitation and housing, etc.